This is Dr. M from Synthesis MedEd. If you're having trouble understanding ketones and ketone formation and how they relate to ketoacidosis states like DKA, then this bite-sized biochem video is for you. In our previous bite-sized biochem videos, we discussed acetyl-CoA as fuel for the TCA cycle, and we also discussed the storage of acetyl-CoA as fatty acids and triglycerides, and I've linked those videos here if you need to review them. But now let's talk about ketones. So what exactly is a ketone, and where does it come from? In chemistry, a ketone is a molecule that contains a carbonyl group within a carbon chain, and a carbonyl group is a carbon atom double bonded to an oxygen atom. In medicine, the ketones we're concerned with are the ones produced during fasting or starvation states. Fasting or starvation states result when there is not enough glucose in the bloodstream to provide the acetyl-CoA necessary for the TCA cycle. The body will release stored energy. The stored energy is found in triglycerides, which are broken down into fatty acids. The fatty acids are then transported through the bloodstream to the liver hepatocytes. Here, the fatty acids are broken down into acetyl-CoA groups via beta oxidation. These acetyl-CoA groups can then be fed into the TCA cycle and used for energy. But the acetyl-CoA groups have to be transported to the tissues of the body to be utilized. And acetyl-CoA cannot be transported in the bloodstream. This is because the acetyl-CoA group is too unstable to be transported, and also because it's not permeable to cell membranes. Because of this, the acetyl-CoA has to be packaged for transport into a stable package, and that stable package is in the form of ketones. So within the hepatocytes, a 2-carbon acetyl-CoA molecule is joined to another 2-carbon acetyl-CoA molecule, and after a few steps, we produce a chemical called acetoacetate, which is a 4-carbon molecule. Remember that the prefix ACET indicates 2 carbons, and that's why this molecule is named acetoacetate. Acetoacetate is then further processed and converted into another 4-carbon molecule called beta-hydroxybutyrate. This molecule gets its name because it is a 4-carbon deprotonated carboxylic acid with a hydroxyl group in the beta carbon position. Both acetoacetate and beta-hydroxybutyrate are relatively stable ketone molecules that can be used to transport acetyl-CoA in the bloodstream to other tissues. Beta-hydroxybutyrate specifically can usually be measured in serum blood tests in most laboratories if ketosis is suspected. Once these ketones arrive at other tissues, they are absorbed, then broken back down into 2 molecules of acetyl-CoA. And the acetyl-CoA can then be directly fed into the TCA cycle to produce energy in the form of ATP. Now, there is one important fact to note about acetoacetate. During transport in the bloodstream, it can undergo a spontaneous reaction called decarboxylation, where it loses 1 carbon in the form of CO2. This leaves a 3-carbon molecule known as acetone. Acetone is highly volatile, which means it evaporates very easily, which means that when this molecule reaches the pulmonary capillaries, it can escape into the lungs as gaseous acetone, and this can then be exhaled and can be detected on the patient's breath as what has been called a fruity odor. Also recall that acetone is one of the main components of nail polish remover, and so that same fruity odor can also be described as a nail polish remover smell. The buildup of acetone can also be measured in the bloodstream, and some laboratories have the capability to measure serum acetone if they don't have the capability to measure serum beta-hydroxybutyrate. Ketone production can have an effect on acid-base balance. The process of ketone formation produces hydrogen ions, and these hydrogen ions can build up and cause acidosis. Now, there is some terminology and physiology that we have to discuss. A buildup of ketones in the body is known as ketosis. Now, ketosis is not necessarily a bad thing. During periods of fasting, when the liver's glycogen stores have been depleted, ketosis will occur as the body releases stored energy from fats. And as long as the excess protons are able to be buffered and eliminated, the body's acid-base balance will not be disturbed. and therefore acidosis will not develop. But if the ketone production continues, then the acid-base balance will be disturbed, hydrogen ions will build up, and acidosis will occur. And at this point, the body enters a condition called ketoacidosis, which is an acidosis resulting from the ketosis. And ketoacidosis is an abnormal condition. Now, how do we identify ketoacidosis? We start with the serum chemistry panel. Ketosis leads to ketoacidosis, which will result in bicarbonate loss due to buffering of hydrogen ion. On the serum chemistry panel, we will see a decrease in the CO2, which is the venous bicarbonate. Ketones also contribute to the anion gap because they are an unmeasured anion. If you don't recall this concept, I've linked a video here describing metabolic acidosis in detail. A decreased bicarbonate level in the setting of an increased anion gap is known as a high anion gap metabolic acidosis. One of the causes of high anion gap metabolic acidosis is ketoacidosis. And so if we suspect this, we can then measure the serum beta-hydroxybutyrate, or if this is unavailable, the serum acetone. And this will help us identify a ketoacidosis. So what can cause ketoacidosis? There are three primary etiologies of ketoacidosis. As we've discussed already, a starvation state can result in a ketosis and a subsequent ketoacidosis. And this can be caused by a decreased intake of nutrients, specifically glucose and carbohydrates. Malnutrition can cause this, and keto diets can also lead to this. A starvation state can also result from too much elimination of nutrients, either from the top or from the bottom. From the top, too much vomiting. From the bottom, excessive diarrhea. A second cause of ketosis and ketoacidosis, which is very common, is diabetic ketoacidosis, in which the breakdown of triglycerides and fatty acids is triggered by insufficient insulin levels in the bloodstream to properly utilize the glucose that is present. And a third cause is alcoholic ketoacidosis related to chronic alcohol use. The specific mechanisms which lead to these three causes of ketosis and ketoacidosis are beyond the scope of this video, but I do recommend that you research these further on your own. So to summarize, ketones are basically packages of acetyl-CoA that are transporting it in the bloodstream to the tissues that require it. A buildup of ketones is called ketosis. Ketone formation leads to acid production, and this will ultimately result in ketoacidosis if not corrected. Ketoacidosis will result in a high anion gap metabolic acidosis, which results in a low bicarbonate and high anion gap on the chemistry panel. And the three main causes of ketoacidosis are starvation ketoacidosis, diabetic ketoacidosis, and alcoholic ketoacidosis. Thanks for tuning in, and remember, don't aim to memorize, aim to understand so that you can then apply what you've learned. See you next time.